Now it is my pleasure to introduce Dr. Alexandra Tinnermann, who will be kicking off day 2 of the spinal cord imaging workshop. Dr. Tinnermann completed her PhD under Christian Buchel in the Department of Systems Neuroscience at the University Medical Center Hamburg-Eppendorf, where she has continued as a postdoctoral research fellow. Dr. Tinnermann's research studies pain, pain modulation, and placebo with brain and spinal cord functional MRI, with several high-impact publications. Her work is especially influential in shaping how we think about pain modulation across the brain and spinal cord axis. Today, Dr. Tinnermann will be discussing task-based functional MRI, which is a topic that I'm highly interested in. Thank you for joining us, Alexandra.
Yeah, thank you so much for inviting me. Very sad that I cannot be there in person. It would be so great to meet you all and be in California, of course. Here it's already dark and very cold. Oh, I will share my screen. Okay. Yeah, and I think I will start. Yeah, so it's my pleasure to talk about task-based fMRI and what you can do with spinal fMRI and experimental paradigms. So just to give you a quick overview, what I will talk about. So I will, as already mentioned, my main focus is pain research. So I will mostly talk about pain, but we recently also had some collaborations with the neurology department. So I will also present some studies that investigated movement or motor-related questions. So with regard to pain, I will mostly talk about pain modulation. I will talk about expectations such as placebo, analgesic treatments, and my last project was on concurrent pain, so 2 different pain stimulations at the same time. And with regard to motor, I will talk about fourth generation and how that compares between healthy subjects and stroke patients. So to give you an overview about the pain system, so when we hurt ourselves, for example, a burn on our hand, a pain signal will travel to the spinal cord, from there to the thalamus, and from there to different cortical regions to generate a pain experience. If we look at that in more detail on the neuronal level, there are specialized neurons, nociceptors, who send this pain signal to the dorsal horn of the spinal cord. And from there, there's an ascending tract that terminates in the thalamus. And from there, the signal goes, for example, to the insular or somatosensory cortex. But apart from this ascending system, we also have a descending pain system or pathway. This one originates in the prefrontal cortex and sends neurons to the brainstem, the periaqueductal gray specifically. And from there are connections to the spinal cord. And if we look at that again in more detail on the neuronal level, there are these descending connections to the periaqueductal gray, and from there are neurons sent to the medulla. And these neurons can both be facilitating or inhibitory, which means that basically the incoming pain signal into the spinal cord can be modulated already here at this level by either inhibiting or facilitating the signal. If we now look at the brain and how pain is processed in the brain, we see that many regions are implicated here. So the most important thing you will notice here, there's no such one pain-specific brain region, but rather it's different regions and networks that are involved to create this pain experience, which are composed of affective components, cognitive or somatosensory, for example. And all this leads to the fact that pain is basically the result of different factors and modulatory processes, such as the context we are in, the mood we are having, the cognitive state, or physiology, of course, also plays a role. All this can influence how painful we perceive a certain painful stimulus. So, although my talk is not technically about combined corticospinal imaging, but most of the studies I did was using a sequence that allowed measuring both responses in the brain and in the spinal cord. And this, of course, allows us to investigate this ascending pathway, but especially, and that's my main focus, this descending pathway to see how that interacts from the prefrontal cortex down to the spinal cord. There will be a talk later about that, just to give you a little oversight here. One sequence that allows to measure both responses in the brain and in the spinal cord was developed by our physicist, Jürgen Finsterbusch. So basically here, in the end, we will have two data sets, one that generates images from the brain, functional images, and functional images from the spinal cord. First study in our institute who applied this sequence looked at pain perception in general, rather a proof of concept study. And here, this is basically a pain cluster in the dorsal horn of the spinal cord. And here the connectivity to the cortex or to the brain was investigated. And we see that, for example, to the brainstem in the periaqueductal gray or for the insula. So there is increased coupling, which are like known regions to be involved in pain perception. And most interestingly here, there was a correlation between perceived painfulness of the stimuli and the coupling strength. So the coupling between the spinal cord and the periaqueductal gray showed a correlation in the sense that people who perceived that as more painful also showed increased coupling in this pathway. So I will mainly talk about three studies now that I conducted using this corticospinal imaging approach to look to study pain modulation and its influence on spinal activity and the corticospinal coupling. And in the first, we asked the question whether negative pain expectations modulate spinal activity and also the coupling along the descending pathway. So I will briefly explain or talk a little bit about these expectations. I'm not sure how familiar you are with placebo and nocebo effects or experimental paradigms. So placebo hypoalgesia is basically the pain decrease that results from pairing a sham treatment with hypoalgesic expectations and experiences. And how we implement that in the lab is usually through a treatment like a cream that is applied to the participant's arm. And as it's a sham treatment, so basically it's just an inert fatty cream, but the information we are giving to the participants differs, of course. So here they would receive the information that one is a control cream, while the other one is a placebo. Well, they don't receive the information as placebo, but it's an analgesic agent that will decrease their pain perception. And then during a learning or conditioning phase, we will actually manipulate temperatures, which is unbeknownst to the participants, to make them believe that this placebo cream really decreases their pain perception. And then during a test phase, they receive identical pain stimuli. And in theory, we would expect that people show then reduced pain in this condition. Nocebo hyperalgesia is the opposite. So here there's a pain increase that results from pairing shunt treatments with hyperalgesic expectations and experiences. And the setup is very similar, just the information that we are giving is different. So in a nocebo condition, for example, the information could be that the cream people are receiving contains capsaicin, which is the hot ingredient of chili peppers, and that naturally increases your pain perception. And here the experience will be reversed. So people will experience more pain in the learning phase under this nocebo condition. And then we would expect, when giving identical stimuli, that here in the nocebo condition, people will report more pain. So there is indirect evidence that this descending pathway is actually able to modulate spinal activity in the context of these expectation experiments. So we don't know the entire pathway, but we know that at the level of the spinal cord, this is also work done by previous colleagues of mine, they showed that in the dorsal horn of the spinal cord, in this placebo condition, there is reduced activity compared to the control condition. And in the opposite, in the nocebo hyperalgesia experiment, people also showed increased activity this time under the nocebo condition, so more activity and more pain reports as well in the nocebo compared to the control condition. And this led to my study, where we built on these effects. So here we invented a cream that doesn't exist, and the cover story was as follows, that this is a cream usually used in atopic dermatitis patients, but that it has a known side effect that it will increase pain sensitivity locally on the skin when applied. And then we created two versions of this cream. One was a cheap version, and the other one a supposedly brand or expensive version. And this is what we intended it to look like or resemble a little bit. At least in Germany, I think this resembles more the cheap version and this the expensive version. And then people were assigned to one of the two creams. And in most of our experiments, we apply heat pain stimuli. So we applied cream here on the arms, and people received heat pain stimuli in a control condition and in this nocebo condition. And when we look at pain perception between groups, we see there's rather not much difference in the cheap group between control and nocebo, but for the expensive nocebo group, we see a clear difference in pain perception, increased pain perception for the nocebo. So of course, the question is, how is this behavioral effect mirrored in the neural system? And first, we were interested, is this effect represented in the spinal cord? And we see a cluster in dorsal areas, but not more medial. But here we see a clear increase in activity in the nocebo condition, but this is across both groups. In the next question, we were interested whether we can also find this interaction effect, so increased activity in the expensive group compared to the cheap group. And here, this is a cluster in the periaqueductal gray that actually showed this, or mirrored this behavioral pattern with a clear increase in activity under the expensive nocebo condition. Then we also looked for correlates with behavioral effects. So basically, every participant has a nocebo magnitude effect, let's say, and we correlated that. And this gave us a cluster in the rostral anterior cingulate cortex, where we find a negative correlation across all participants, showing that people who have larger nocebo effects show reduced activity in the prefrontal cortex. And lastly, we were interested to see whether we can find changes in coupling along this descending pathway. And therefore, we extract the time courses or the seed region in the spinal cord and also here in the prefrontal cortex. And interestingly, both of these seed regions showed coupling differences with the periaqueductal gray. And this is a little complicated, but there's actually a correlation between the behavioral effects and the coupling strength. And that differs between groups. So we see here the expensive nocebo group shows a rather negative correlation, and the cheap group shows a positive correlation in coupling strength, depending on the behavioral effect they are showing. And this, to make it even more complicated, this effect is then reversed between the spinal cord and then the PAG. So here it's the expensive group that shows increased coupling with larger nocebo effects. So concluding here, we can say, yes, negative pain expectations modulate spinal activity and also the coupling along the descending pathway. And in the next study, we looked at opioids. So this is has also an expectation component, but we were actually interested in the interplay between drug or analgesic treatments and expectations. And just to give you an overview, so this is basically where a lot of opioid receptors are located in the brain and also in the spinal cord. And if you remember the brain regions involved in pain that I showed you, you might remember that many of these regions that are involved in pain perception also show high density of opioid receptors, and also here the dorsal horn of the spinal cord. And interestingly, this expectancy component is actually showing that when we give analgesic treatments, the analgesic effect itself is always a combination of the agent itself, but also other, like for example, expectancy effects. So this can be clearly seen here. Compared to a baseline condition with no opioid, people receive an opioid while lying in the scanner actually, and they're not aware that they're receiving the opioid at this moment. So there you see a slight pain decrease. But if you then tell people, now we will start the opioid treatment, and you just continue giving the opioid treatment, you will see a further pain decrease, showing that the analgesic response we are showing often also incorporates expectancy effects. So in the study design, we wanted to investigate whether the level of knowledge or certainty also influences analgesic responses. So here we had 3 different groups, one that received saline and one that had a 50% chance of receiving remifentanil. So they were explicitly told that, and they were blinded. And in this group, people were not blinded, and we told them, you will definitely receive remifentanil. Here we just started with a baseline phase without any infusion, and then in the middle of the experiment, the infusion started with either saline or remifentanil, then composing the treatment phase. And here again, we used heat pain stimuli on the arm. With regard to the behavioral effects, in this study, we can conclude that the certainty did not modulate opioid analgesia. So we see here that the saline group shows basically a difference around 0, so no analgesia, while both opioid groups show clear analgesia, but no difference between these two groups, which might have one reason might have been that I think people were mostly unblinded very easily because they felt that they received remifentanil. So that, yeah, makes it a little difficult to tell whether it was certainty or uncertainty in this group. So here we pooled both remifentanil groups for the rest of the analysis. And first looked whether we find pain decreases in the brain as you would expect under remifentanil. And here this is typical pain-related brain regions that show a clear decrease in activity under remifentanil. And we repeated this analysis in the spinal cord. And here we actually see in the dorsal horn, and this is of course always important that we are ipsilateral to the stimulation site, and this is the case here, we find increased activity under remifentanil. Then we looked again at regions of the pain modulatory system and whether we can find, for example, correlations with behavioral effects. And here we find a negative correlation between analgesia and activity in the periaqueductal gray. So the more analgesia people showed, the more reduced was activity in this brain region. And we found the opposite pattern in the prefrontal cortex, in the VMPFC, for example, where there's a positive correlation between analgesia, showing that more analgesia resulted in increased activity in this brain region. And finally, we also wanted to see whether the coupling was modulated by the opioids. And here, as in like comparison to the other study, we also had a seed region in the prefrontal cortex and in the spinal cord. And well, to my surprise, actually, we found a similar result that there was coupling that was influenced by the analgesic level that people experienced. So between the prefrontal cortex and the PAD, we find this correlation pattern between the saline and the remifentanil group, and similarly between the spinal cord and this region in the periaqueductal gray, we find a correlation pattern between saline and remifentanil that is also reversed or opposite. So here I would conclude that opioids also modulate spinal activity and coupling along descending pathways. And that brings me to the last pain study where we looked at concurrent pain and whether that also modulates spinal activity and coupling along the descending pathway. And what do I mean by concurrent pain? So this is actually an idea or a paradigm from the pain community, which is called conditioned pain modulation. And this consists of one longer lasting painful stimulus, and you then apply second shorter stimuli either at the same time or shortly after applying this long stimulus. And this is a paradigm or finding that's often called pain inhibits pain. And why is that the case? Because when we just apply this stimulus alone, we have a certain pain perception, but when it's paired with another painful stimulus, we see a pain reduction. So here in this study, we used a different pain modality. This was a pressure cuff, actually, and that was here on the arm. So there was a tonic, long-lasting stimulus, and on the other arm, shorter phasic stimuli were applied. And to give a overview about the stimulus types applied in this study. So we had a control condition where basically the pressure cuff was inflated but not painful. That's why it's here around the 0 line. And in between, shorter phasic stimuli were applied. In the CPM conditions, so condition pain modulation condition, the tonic stimulus was painful, and in between, shorter phasic stimuli were applied. So here in this study, we didn't find mean differences between the CPM and control condition, but rather a steady decrease over time. So apparently here the CPM resulted in a decrease with time in pain intensity. Yeah, and well, I'm not sure whether this is due to the pressure stimuli that were used in the study, but actually it's a very rich data set because so here you see regions that show differences or respond to pain stimuli. And the interesting thing here is that I looked at time courses of both responses. And actually for the heat pain studies, I was actually never able to extract these time courses because the signal was too noisy. And here that is not the case. So I extracted time courses from different regions, also here from the dorsal horn. And as you can see in gray here, this is a response to the pressure stimuli. And I think it runs a little against current hypotheses that say that the bolt or the HRF function does not differ between the spinal cord and the brain. But I would argue that at least here for pressure pain, there's a very fast increase in the bolt response that would be not expected when using a normal HRF function. So this is here in black, the stimulus convolved with the HRF. So that would predict a way later signal. So because of these discrepancies, actually, we stayed with time course analyses in the study. So here first we looked whether we can find brain regions that show decreased activity under the CPM condition. And you see, when we look at these time courses, there are many regions known to be involved in pain perception that show a decrease in the CPM condition. The same is true for brainstem regions. So for example, here in the medulla, we similarly see that the pain signal is reduced under the CPM condition. And again, in the spinal cord, we see here that the CPM signal is reduced compared to the control condition. And this is again in the dorsal horn, this time on the right side, because the phasic short stimuli were on the right arm. Finally, we also ran some connectivity analyses. So here, this is not easily transferable from the other two studies. So here, we just have a connectivity with the seat region in the spinal cord. And this shows increased coupling under the CPM condition to regions of the descending pain system, such as the medulla or prefrontal regions, such as the anterior cingulate cortex or the VMPSC. So with that, I would conclude that concurrent pain stimulation also modulates spinal activity and coupling along descending pain pathways. And just a short summary for these pain studies. So pain modulatory behavioral responses are actually represented in the spinal cord or mirrored. Hypoalgesia is represented through increased coupling along this corticospinal increased along this descending pain pathway. For hypoalgesia, the pattern was, well, not so clear, I would say. So here again, it is represented somehow in coupling along this descending pathway, but here I would not conclude whether coupling is increased or decreased. But we see that these pain modulatory systems somehow modulate the coupling along these regions. So that brings me now to the motor part of this talk. So the motor system, of course, is also interesting because we have a direct connection from the motor cortex to the spinal cord. And from there, muscles are targeted that allow us to move. If we look at that on the neural or neuronal level, here is a direct connection from the primary motor cortex to the ventral horn of the spinal cord, and that basically sends a neuron to the muscle. So here I will present two short studies. So the first question was whether the spinal cord mirrors force levels, different force levels that are known to be represented in the motor cortex, for example. And in the second study, we will see how that modulates, or how these signals are modulated under stroke. So the study design is quite simple. People just generate force with their hands with this device, and there are three different levels of intensity: 30, 50, 70%. And the design itself is that the intensity level is signaled to the participants, and then for 15 seconds they have to press the force level that is required, and this circle shows them whether they are around the target force level. And for the brain, we see motor-related activity in primary cortices and so on. And here we actually see a parametric response, so increasing activity with increasing force level. And the ventral horn of the spinal cord similarly shows this increase with force level. So here we have low, medium, high, and an increasing activity level in the spinal cord. And since they both show this pattern, it's maybe not surprising that there's also a correlation between the signal in M1 and the signal in the spinal cord. So here we can conclude that the spinal cord mirrors the responses in the primary motor cortex. And now we want to see how that is changed under stroke. So here again, the stimulus design was identical, but this time stroke patients were recruited. They all had the stroke in the same hemisphere, spanning, for example, the motor areas and also a little down to the insula. And in general, what was observed in this study, that there was slightly increased activity in the spinal cord, but also in some cortical regions, mostly motor planning or secondary motor areas, where stroke patients showed increased activity. And interestingly here, we also investigated the coupling strength between motor areas and how that relates to motor impairment. So between the supplementary motor area and the ventral horn of the spinal cord, there was coupling occurring. And similarly, between the ventral premotor cortex and more dorsal areas in the spinal cord, there was also coupling occurring. And now the interesting bit is that this coupling correlated with the degree of motor impairment in these patients. So here, basically, these are people with stronger motor impairment, and they show reduced coupling in this connection. And in opposite, in this connection, the coupling showed the reversed pattern. So people who had more motor impairment here showed increased coupling in this connection. Yeah, so with that, we can conclude that under stroke, the coupling is modulated between the promotor cortex and the spinal cord. As a summary, I would say that the degree of motor impairment in stroke patients corresponds to the coupling strength in the motor system. And a more general summary, summarizing all studies now, is that spinal or corticospinal fMRI is a feasible tool to investigate pain and motor-related processes. And here, pain and motor-related task designs can reveal spinal and corticospinal mechanisms in pain modulation and motor impairment. And that brings me now to the last bit of this talk. So I was asked to share some personal experiences about spinal fMRI and probably also task-based fMRI. So the first question that I was asked what I think are the key problems or pitfalls, and I think that's still the registration or normalization of the spinal data. So I think the first study with the nocebo effects, I actually co-registered images manually because the algorithms that existed at the time weren't good enough. So that was a lot of work. And in terms of reproducibility, it's obviously not a good idea. So what has worked well in that sense is that the development of the spinal cord toolbox marked a major improvement in the pre-processing of the spinal images so far. But that brings me also to the last point, what we can do better as a field. So the spinal cord toolbox, especially these normalization and registration algorithms, offer a very wide variety of parameters. That can be changed and adapted. And so far, I have tried to find parameters for every participant that kind of create a good or result in a good normalization. And I think that maybe also not the best approach because it's also very time consuming trying out so many different parameter settings. So what I think would be a good idea, what we could do as a field, especially for those working with functional fMRI, is to establish standardized pre-processing pipelines where I would point to the most important part is finding parameters that are robust and work for many studies and for many subjects. Because that was also one of the pitfalls I fell into, is once I analyzed one data set with the spinal cord toolbox and I had a new data set, I thought I could just use the same parameters for the new data set, but that didn't work well, to be honest. So I had to find new parameters for the new data set. And I'm not sure right now. I found some parameters that at least worked well in 2 other data sets so far. But I think a lot of testing would be needed to have a final conclusion here. But yeah, I would think that would be a great idea to share experiences also between research sites and to see what kind of parameters, for example, have worked for others. And then I also have some practical tips and recommendations. So I would always suggest to have fewer, but therefore longer runs. So the opioid studies, for medical reasons, we had very short runs, but therefore 8 runs in total. And I think people tend to move more between runs. So this is of course not good for data quality. And it's always important to have more trials for spinal studies than for brain studies. And in the last study, so with the conditioned pain modulation, I implemented white matter regressors. So I've seen that in other preprints or papers as well, that this is now done. And I can just recommend it. So I think it increased the data quality a lot. So maybe if someone's interested, I can share later on how I extracted those regressors. And I think another important point is piloting. So it's important to keep in mind that when you've run a study in the brain and used an experimental paradigm or stimulation equipment, this will not necessarily mean this will work in the spinal cord. So I, for example, had a study where we used this heat pain stimulation device, and people never, so basically they were calibrated, but then once the scanner started, they always said the pain was not painful. So we cannot test this equipment in the scanner, so it's not entirely sure that it's due to the stimulation device itself, but well, I just will make you more sensible to that. So basically, because in spinal or corticospinal fMRI, people tend to lie deeper in the bore of the scanner. So there are different physical properties working on the equipment you're using. So you should keep that in mind and test that. Then there might be differences between pain modalities, for example. So as I said, with the pressure pain, I was able to see time courses now, which was not possible with heat. And now we're running a study with electrical stimuli, for example, so there's no time courses, but they also show very nice activation in the spinal cord. And my experience is with heat, it's often very difficult to even get a proper group result. And then the voxel size, you should consider that. So in the last study that I did, we used a larger voxel size. So here we had 1.2 times 5 millimeters. So this was basically combining the worst of the other settings we used before, but I'm not sure, maybe that was also one of the reasons why I have a better signal now, but I think it can be worth piloting or even comparing between studies. And then for the movement, for example, it also makes a difference whether you really just hold or whether you have repetitive slow movements. So in the fourth task that I presented, I said it was fourth generation, but actually it was not isometric throughout these 15 seconds, but people were instructed to basically do that in a repetitive way, because my colleague who ran the study actually piloted that and found that these repetitive movements create way stronger activity in the spinal cord. And with that, I would like to thank my co-authors. So this is my colleague Hannah Braas, who ran the studies in the stroke patients. And this is our physicist, Jürgen Finsterbusch. Yeah, and with that, I like to thank you for listening to my talk. And I'm curious for your questions and the discussion.
Thank you so much for that awesome talk. We will start with opening up questions to those in-person attendees. And we'll start with Christine. She was first up.
Hi, thank you for the talk. I'm Christine, big fan of your work for years. So one questions I have is, why do you think you see the time course from the pressure pain, but not the thermal pain?
Well, one explanation could be just the area that's being stimulated. So if you use, so let's see if you see me, but if you have the pressure cuff that's around the entire arm, so it's a lot of skin area basically that's being stimulated. So probably way more neurons. And the heat device is just, so I think it's just a 3 by 3 centimeter area that's being stimulated. So this could be one reason that it's just too small, the area that's been stimulated.
I see. Second question is, earlier you talk about placebo and nocebo. Do you expect the spinal cord also show signals when simply when we are anticipating nauseous stimulus coming on? In other words, if we tell our participants, well, the pain is coming, will you expect their spinal cord activation?
I think if I can remember that correctly, so for the nocebo study, I actually looked at the Q phase, and I think there was a tiny cluster coming up. I'm not sure if it was for pain in general or even for nocebo. If I remember correctly, it might have been even increased activity for nocebo expectancy, basically. But that was a very tiny cluster, so not significant. But maybe if we would have more participants and maybe stronger effects, we would definitely see anticipation effects, I guess.
Thank you.
All right. Moving over here.
Hi, it was a fabulous talk. I have a couple of questions. First of all, I would like to know what's your observation and suggestion regarding what HRF to use for spinal cords related to, let's say, pain or any other tasks. Should they be different, or should we look into different HRFs than the conventional ones that we use? Yeah, the first one.
Yeah, so far I haven't used other HRS because if I remember correctly, there was a monkey study actually investigating HRFs in the spinal cord, and they concluded that they're quite similar to HRS in the brain. That's why we kept with these HRF models. I mean, maybe I wouldn't change HRS based on one study, but maybe if other studies would similarly find different time courses, it might be worth considering other HRS or just sticking with time course analyses. I'm a big fan of that anyway, so maybe then it makes more sense to just directly look at these effects because they do not assume any model behind it.
Yeah. Thanks. The other question is related to the results from the stroke study, in which you computed the connectivity between brain and the spinal cord. Specific regions you showed, like for instance, I think the motor cortex with the ventral horns and the premotor cortex with the dorsal horns. I was curious to know that in that study, did you consider the correlations between other regions too? Like, for example, did you consider what's the connectivity of the motor cortex with, let's say, the dorsal horns of the spinal cord or in other parts of the spinal cord?
That's a good question. I think, no, I think that's basically what the analysis was focused on.
Yeah, I think because the connections from the motor cortex comes down to the ventral horns through the corticospinal tracts. That's the usual way of investigations. But I was just curious whether you looked into other connections also.
No, I think that's it. I mean, from the so there was also dorsal horn activity, which is rather surprising, right? But that basically we interpreted as interneurons that are targeted here, and it must be clearly be another effect because also the connectivity was inverted, or the correlation between motor impairment and coupling strength. So this hints to the fact that it's not the direct influence from the motor cortex onto the ventral horn.
Yeah, thanks a lot for your answers.
Yeah, great question. I think the hemodynamics response function question always comes up, and it's something that we should come together and investigate because we just stick with the default, and because it's the default, and we don't have a strong reason not to use it. So quite exciting. And then also kind of the descending motor commands I think are actually quite interesting. And it's much more than just that monosynaptic connection to the motor neurons. And there's a lot of other modulation coming down that can affect what comes up and what's happening in the cord. So just complex systems. Next question. Oh, 3. I don't know. Yeah. Merve's in charge. She gets priority.
I am reading a question on behalf of one of our attendees, Caroline Landel. So I'm going to read it word by word. Thank you, Alexandra, for the very clear presentation. Do you think that using two distinct fields of view for the brain and spinal cord could sometimes introduce temporal or spatial distortion in the signals between these two structures, potentially explaining why is it sometimes hard to interpret the direction of brain and spinal cord couplings?
The couplings, so usually the data quality is a little worse, especially in the spinal cord when you combine these measurements. So that you have to take into account that on top of the normal lower signal to noise in the spinal cord, this will introduce even more noise. But with regard to the coupling, of course, I mean, this is basically correlational analysis, so we can never know the direction of these coupling. It's rather more from basically an anatomical perspective. Maybe you could conclude whether it's a descending or an ascending coupling. So, for example, for the motor cortex, I think most people would agree that must be a descending signal, right? For pain, it's not always clear. I mean, especially the connection between the spinal cord and the brain stem, they are also ascending connections. So this could also be an ascending signal. That's basically the disadvantage of correlational analysis, fMRI. I mean, there are other ones that could be applied to say more about the directionality, but I think the spinal cord would be way too noisy to actually use them.
Excellent, thank you. And just to comment on that, it's that we have a pretty good sized data set with brain and spinal cord resting state time series. And we'll talk about this later, but we're perplexed by just some of our ability to look at connectivity. And we're getting findings that, I don't know, aren't the strongest that we'd expected. And it'd be fun to open up our data set and everybody come together and look at how different processing pipelines kind of can affect the results. And just a note, I'd like to talk more about that.
Hi, Alexandra. This is Dario. Fantastic talk. I have two quick questions. One is a follow-up to what Caroline asked with correlation between your brain and spinal cord time courses. Can you remind me, did you do whole time course correlations, or did you also try some PPI, maybe for the CPM study that is in preparation?
So these are all PPI analyses, but I think you can still, I would still say they're based on correlation. So this is basically the beta image derived from the PPI analysis, and that I correlate with behavioral responses. So for the CPM study, there wasn't, well, not that clear behavioral effect. So I tried to correlate that as well, but yeah, that didn't show any promising results. So here it's just a pure PPI result.
Okay, got it. Thank you. The second one is about your practical tip of using fewer and longer runs. Can you elaborate a bit on that? Because I've talked to different people. Some prefer shorter and more runs and have better results with that versus fewer and longer.
Okay.
Yeah.
Well, then, yeah, I would be curious to know why they experience is that it's better to have more runs. But then, I don't know. I mean, it was a very long experiment, and people at some point were really suffering. And I think the people who got the opioid felt better once they started receiving the drug. So then their suffering was over in a way. But the people who received saline, poor them. So they were suffering until the end. And my impression just was that during the runs, there was way more movement. So especially in that direction, people were just moving a little bit. And then in the end, that affected data quality. And yeah, that of course then affects the pre-processing and so on. So that was my experience. But if people have other experiences or good experience with shorter runs, then maybe the experiment was not that long in total, or yeah, I don't know.
Okay, thanks.
Quick questions, yeah. 1 minute question.
Hi, Alexandra. This is Julien. Thank you very much for your talk. I had two questions, but I'm only allowed one, I guess. It's more of a tech question. So in the end, you talked about the one of the wish is to standardize the pre-processing pipeline. But then you also say that when you receive new data, you had to change some parameters. So I guess do you think it's possible to have really like a standard notebook where that everybody can use with their data? And also I'm curious to know what are the parameters you had to change in the processing pipeline.
Well, the reason why I had to change them, I actually don't know. So, I mean, that was a different scanner. So the first study was still a Trio, and now we have a Prisma. So probably that has a large influence already. But yeah, that was actually my experience that even in one participant, for example, if you change the co-registration slightly between the APIs and the T2 image, a native space, and then do the normalization, it could be or happened to me that basically I needed other parameters for the same participant after just slightly changing this registration step in native space. So I found that it might be very sensitive to just minor changes, but maybe that were just the best or optimal parameters, because now actually I have this data set. I applied that to a new data set that seemed to work fine. So I haven't looked at all participants yet, but at the participants that I looked at, they all looked very well, the normalization results. And I provided these parameters to a colleague with electrical stimulation, And she said it didn't work for all participants, but for most. Yeah.
All right, thank you. Thank you very much, Alexandra, and wonderful presentation. You probably want to sleep, but if you do want to wake up in a few hours and join the panel discussion, you're more than welcome.